All right. Hello, everyone. According to my clock, it's now a couple minutes after, so we're going to get started while some more people join. My name is Jeff Stallman. I am the Fungus Conservation Coordinator at the Global Center for Species Survival at the Indianapolis Zoo. That's who's hosting this webinar in honor of Reverse the Red Day. So on Reverse the Red Day, we celebrate the conservation of biodiversity and the actions that we're taking globally for species conservation in a bunch of different forms. And I think today we have three great examples of that. So we have three speakers who all have some connection to the IUCN-SSC, so that's the International Union for the Conservation of Nature, the Species Survival Commission, and the fungal groups, or the groups that focus on fungi specifically within this network. So we're going to hear from Josefa Raja, Kelmer Martins Cunha, and Greg Mueller in this order. And the general format will be each speaker will speak for around 15 minutes. And if we have time within that 15 minutes, we'll take questions for that presentation. If not, we'll revisit it at the end when we have, 10 or 15 minutes for open discussion. So I'd recommend putting your questions in the Q&A field. I'll also try to monitor the chat, but that's probably the best way to do it. And likely at the end, we can open it up for you to voice your questions as well if you prefer to speak them. So with that said, we are going to start today with Dr. Josefa Raja, who is a research scientist at the University of North Carolina at Greensboro and a member of the IUCN SSC Aquatic Fungi Specialist Group. So Josefa, please go ahead when you're ready.
Yes, one second. Let me just All right. Thank you very much, Jeff. I appreciate the invite, and thank you everyone for joining today. It's my great pleasure to be here today to talk about the work that I've done in conjunction with a grant that I received from the Indianapolis Zoo, without which I wouldn't be able to talk about this fungus Atromagnispora, which was newly described recently from Indiana. One second. So, aquatic fungi are really rising up in the conservation front. In the last 3 to 4 years, several things have happened that have allowed aquatic fungi to be sort of on the rising front of conservation. Basically, it started all with Moni Bohm, who's a freshwater conservationist at Indianapolis Zoo, contacting me, and we did this magazine with freshwater fungi and fantastic freshwater species. We sort of highlighted some of the cool species in freshwater habitats. And then in Europe, there have been a couple of things happening. Two important projects, Fun Action and Most Fun, that have been going on that are absolutely fantastic, looking at conservation, discovery, assessment, monitoring of freshwater fungi in Europe. And finally, in 2023, in October, we formed the first aquatic fungal specialist group. And that was very cool because there was no such group before. And you can see in the picture here, Moni Bohm with the chair and the co-chair, Isabel and Sally Fryer, that are basically running the group. And right now, I think we have about 25 to 30 or so members, and it's growing. So it's been really a great time to be in aquatic fungi. And basically, all of this work started for me when I first met Moni Baum, and we wrote this little grant that was sort of funded by the Indianapolis Zoo, the Global Species Survival Center. And I was interested in aquatic fungi, so we wrote this grant and got funded, and the story begins, essentially. And the title of the grant was What's in the Water? Discovering the Freshwater Ascomycetes Fungi of Indiana. And at that time, we were focused on Marion County, but right now I'm trying to get fungal samples from all over the state of Indiana. I've been lucky to meet several interesting scientists at the Indianapolis Zoo and at the Global Species Survival Center. And it's amazing how all of these guys are so interested in conservation. It's really exciting to be a part of this conservation movement. And I met some great people there, including Moni and Jeff and Justin and Andrea and Samuel and Corel. So it was really a lot of fun. I also met Katia and Sergio, who are not at the zoo anymore, but it was really exciting and interesting to meet all of these amazing scientists, all with the main goal of trying to conserve organisms. And so basically with that, I'm going to get into what I'm doing with the freshwater ascomycetes. These are fungi that are basically occurring in aquatic habitats, in freshwater habitats, and they occur on submerged or partially submerged substrates in these habitats, right? And they can occur on wood or leaves, and different forms are seen on different substrates. Primarily, the sexual forms are seen on the wood, and the asexual forms are seen on leaves. And so you can see here pictures of myosporic, the sexual structures seen on submerged wood. On the left, you can see fruiting bodies of these fungi on the wood. And then once you open up the fruiting bodies, you kind of get spores, asci and ascospores. And you can see some of the pictures of some of the very interesting fungi in this group, which is an ecological group, not a taxonomic group. Then here are some of these asexual forms that you see in the water as well. They are aquatic hyphomycetes that are variously shaped. You have the aeroaquatic hyphomycetes that are also very interesting. There are about 300 species of aquatic hyphomycetes, close to 80 species of aeroaquatic hyphomycetes. The aeroaquatic hyphomycetes are very cool because they occur in habitats which are standing bodies of water, right? And so when the substrate on which they're growing sort of come up at the air-water interface, these little propagules of spores detach and they go and find new substrates. And then in freshwater habitats, you find these other mitosporic fungi, which are like a cornucopia of all kinds of terrestrial species that have found their way and sort of evolved in freshwater habitats. So that's the general sort of ecological context of the different groups that you see in freshwater habitats. So how do we find them? The ones that I'm studying, you have to go to the stream, a lake or a bog or a swamp. You go collect submerged material, bring them back to the laboratory, and incubate the wood. And they're mostly seen near the littoral zones of lakes and very close to the shore where the riparian vegetation is overhanging and it drops the substrates into the water. They decompose and they cause a rot called as a soft rot. And so what I do is I bring these substrates back to the laboratory, right, and incubate them in boxes, and then periodically look for fungal fruiting bodies. And when I see those fruiting bodies, I eventually isolate them in pure culture using antibiotic water agar. We use antibiotics because there's a lot of bacteria in aquatic habitats, and so that kind of takes care of the bacteria. So we can only get the fungus to grow, and then we get pure fungal cultures that are exenic, and we can do our work with them. So I've been studying these aquatic fungi in Indiana now since 2024. We've done about 2 different collections. Total of 37 collections are made. This is just a map of where some of the collections are made throughout the state of Indiana. But you can see a lot of them are focused close to Indianapolis and the whereabouts. But also, I've been fortunate enough to collaborate with Dr. Luke, who's basically helped me collect samples. He's been studying the mayflies, which are also aquatic in nature, and he's been very kind to send me samples of submerged wood when he goes out and collects his mayflies. So I have like approximately 40 or so collections now from different places in Indiana, which I haven't been able to go to all of them. So it's fantastic to have Luke, who also has this grant, like I said, to study mayflies from the Indianapolis Zoo. So basically, here you can see this is the place where we first found the fungus, and essentially it is on the White River. So the east fork of the White River, which actually also goes through behind the Indianapolis Zoo. So that was where Luke and both Sergio helped me with collection of these samples. They sent me the submerged wood that they collected. They were looking for mayflies, and they sent me some samples, and I got the samples to my laboratory, and that's where I kind of started looking through those samples and found this interesting fungus that I'm about to talk more about. And here is a close-up of the habitat where the fungus was found. You can see that's where the submerged wood is lying decomposed in the water. And then once brought to the laboratory, this is the wood that is being incubated here. and that's the sample that I was looking at, and I found Atromagnispora. So how does it look like? This is how the fungus looks like. Basically, it is a sexual form of the ascomycete. So it makes ascospores and asci. And you can see this is the wood, and then you can see the fruiting body and these really dark black spores that it makes, which was the reason why I call it Atromagnispora, because it's this dark spored fungus. And so basically I got it in pure culture. And then these are more pictures of this beautiful fungus. You can see on this growing on submerged wood here. This is actually, the wood is now not submerged anymore, but it's fruiting on wood. It makes very thick walled asci, which are globose to subglobose. But the interesting thing is the fungus is not dark always. It starts out as being really light in color, hyaline in color, and it has this really interesting two-pronged sheath. One is this striated sheath, and then this other gelatinous sheath right here, which eventually starts to compress, and the spore starts to get more pigmented. And then overall, at the end, it becomes really dark colored and brown, and it's also rough walled, basically. So the fungus actually reminds me of various other species that have been described from both freshwater and terrestrial habitats. And one of them is Cariospora. So if you look at the spore of this fungus and this fungus, they look very identical. So I was thinking this fungus probably belongs to the genus Cariospora. But as you look more into the literature, you find several other species from freshwater habitats that are similar. One of them is Frigidispora. The other one is Angustispora from Egypt. Frigidispora is from the UK. And then obviously this genus Zopfia, which is also sometimes seen in freshwater habitats, but mostly in terrestrial habitats. So all of these spores have this dark colored. And when I found the fungus first, I thought it could belong to any of these genera. So what do? Well, molecular phylogenetics to the rescue, because if we did not have molecular data, it would be really impossible for me to place this fungus where I eventually placed it. So finally, after sequencing some genes that we use for fungal taxonomy, in this case, the ITS region and the translation elongation factor alpha, which is a single copy protein coding gene, I was able to place this fungus Atromagnispora in the family Lingomycetaceae. Lingomycetaceae is a family consists of primary freshwater fungi, but also some terrestrial species, but mostly on submerged wood and some from other plant material. What I want to point out is that I've been studying this fungus for quite a while, this family for quite a while now. In my grad school, I described some species in this family. And then after I moved from my grad school to my current position, I've also found a few times I found some species in this family Lingomycetaceae. So I was able to place this fungus in the family Lingomycetaceae. However, for me, it was really perplexing. What about the other species? How are they related to this fungus? And so what I did was I sequenced our fungus that I found, Atromagnispora, and then sort of analyzed it with all the other morphologically similar strains or genera. And you can see here, that's where Atromagnispora goes. That's where Angustispora nilensis is. This is where Cariospora is. And this is where Zopfia is. So they're all Dothidiomycetes. They're all bitunicate. But because of convergent evolution, these fungi do not share a common ancestry. They're actually just the spore form is perhaps really useful in aquatic habitats. And one of my hypotheses is that this dark colored spores just float in the water, and then finally they can find a substrate, and that's how the life cycle continues. And so at my current position, I'm also interested in chemistry. So we are interested in analyzing these fungal strains for small molecules, trying to find bioactive compounds that are essentially either cytotoxic, antibiotics, or anti-malarial compounds. And so we have two-step approach here, where we first look at the chemistry, get the strain, isolate the strain, identify the strain, and then grow the strain in large cultures, where then the chemists can take those strains and then look at the chemistry. So they do simple chemical extractions using organic solvents, take the extract and separate it using HPLC. So different peaks of chemicals are separated, and then you can identify the structures either by mass spectrometry or by NMR to in silico sort of identify the novel structure. With respect to Atromagnespora, we have also been doing that. We have a PhD student currently in the lab that is interested in working with this fungus. And here you can see on the left side is a phylogeny, and that's where Atromagnespora is found, and sort of belongs. And then what we do is basically we take all the mass spec data and make a global sort of alignment of all the chemical compounds. So each dot here is a chemical compound. And you can see here that this fungus actually makes interesting cyclodepsipeptides. These are compounds that are made by NRPS systems, non-ribosomal peptide synthetases. And essentially, these compounds, we're trying to find out what their activity are. So these compounds were also described or isolated from a culture called Lingomyces carolinensis before. So this sort of also gave me assurance that, yes, this fungus does belong in the family Lingomycetaceae because of this shared chemical ancestry here. They're related with the chemistry as well. So chemotaxonomy, if you will. And we've also found some new molecules. So the ones in red are some new molecules that have not been described before. So this work is still ongoing. We haven't yet finished all the work on the chemistry side. We want to try to get these compounds, identify them, and test them with cancer assays. We're interested in finding a novel compound that can kill cancer cells, so cytotoxic compounds against breast cancer, ovarian cancer, and melanoma. So this work is still ongoing, but we are also interested in finding new chemical compounds from all the fungi that we find, both from freshwater and from other habitats. And I just wanted to point out that it's a great time to be in the aquatic fungi because I'm co-chairing a session in the upcoming fungal genetics meeting. And you can see we have a session on marine and freshwater fungi, life in water. I think this is the first time that the Fungal Genetics Conference has such a session. So it's great to be able to participate in this. And this is my co-chair. And also one of our members of our aquatic fungal species group, Jennifer Anderson, Dr. Jennifer Anderson, who is going to be a keynote or invited speaker in this session. And her talk is on tetracladium, which she's been studying for the last several years. And the title of that is Benthic and Beyond, Genomic, Biogeographic, and Experimental Insights into the Aquatic and Terrestrial Focus of Tetracladium, essentially. So this is going to be a good conference, the first time where the aquatic fungi are highlighted. Finally, I want to acknowledge the funding that I got from the Indianapolis Zoo. Thanks to Sergio and Luke for helping me find this sample with a new genus. I want to thank the Aquatic Fungal Specialist Group as I'm a member there, and I've learned a lot from all the folks there. And I work in Nick Oberle's lab at UNCG, so he's the chemist that I work with, and we're looking at the chemistry. So I thank him for sort of allowing me to do taxonomy and systematics with him. And finally, my undergrads, who are always helping me culture fungi and look at fungal species. And I want to leave you guys with this sort of thought that the relationship between discovery and conservation is indeed intimate and fundamental, acting as a cycle where understanding the natural world is a prerequisite for protecting it, while conservation efforts also lead to new scientific discoveries. And finally, I would like to thank you for your time and be happy to take any questions if there is any time left. Thank you very much.
That was great. Thank you so much, Josefa. I think in the interest of time, we should probably move on to the next speaker, but we can definitely have questions at the end. So everyone can think of those for the end or enter them in the Q&A now, and we can address them at the end. That was really cool to see the representation of aquatic fungi and amazing micrographs and macro photography as well. It's
Thank you.
Very cool looking fungus. Okay, so we're going to move on to Mr. Kelmer Martins Cunha, who is a master's student at the Federal University of Santa Catarina in Brazil and a member of the IUCN SSC Brazil Fungal Specialist Group. So go ahead when you are ready, Kelmer.
Thank you, Jeff, for the introduction. It is a pleasure to be here with you all in this Robust Red Day to talk a bit about the recent advancements that we had in fungal conservation that are really changing the way in which Brazil's approaches conservation. So I would like to, well, as Jeff said, I'm currently in my master's and I have been involved in fungal ecology and conservation for the past 5 years. So I would really like to state that all of the advancements that you showed to you here today are really are results from a high collaborative efforts among several Brazilian mycologists and mycologists from abroad. So it is a pleasure to be presenting here today, representing all of these advancements for you today. But I would like really to thank all the people that have been concerned with the future of fungi, really acting to preserve it. So fungal conservation in Brazil really started to get traction in the first IUCN Latin America workshop, which was held by Fungi Foundation with the help of the mushroom bracketed and puffball specialist group back then in 2020. And back then, 30 species from Brazil were assessed, so really had none or almost none species assessed for Brazil, and we were already able to include 30 species in the global IUCN Red List. 2 years later, the second workshop was held now in Argentina, also by Fungi Foundation, but now in collaboration with my supervisor, Lisandro Ricardo Dreschler Santos, which because of some experience that we had in the meantime assessing Brazilian fungi species, we actually became, me and Ricardo, official trainers from the Red List unit. And in the meantime, we had also held the first IUCN Brazil workshop. And then in 2023, we held the third Latin American workshop in Panama. I also had the pleasure to be there and help to co-organize it among several other mycologists from Latin America. And then in 2025, we had the second and third IUCN workshops. So really in a short time frame, in 5 years, we started from non-species from Brazil assessed in the global red list to 30 species and now up to 123 species listed in the global red list. This may not seem a huge number, but anyone who is involved in red listing species know that it is a huge workload to do so. And this also represents around 10% of the total fungal species included in the red list. So Brazil is really actively contributing to our understanding of how threatened fungal species are in general. So currently we have 319 species assessed for South America in general, and around 40% of these occurs in Brazil. So the country, along with Argentina, Colombia, Chile, is among the countries with the highest number of species assessed. And as I said, this may be a small number compared to the estimate of total known species for Brazil. But really, these initial assessments gave us the evidence that we needed to really make sure to say that fungal species in our country were really being threatened and facing the same pressures that other groups, such as animal and plants, were facing. So anthropogenic pressures such as agroindustry, urban development, habitat shift and alteration, and of course the interactions with climate change are constantly putting fungal species in our country in an endangered position. So although the current number of Brazilian fungi species included in the global red list is really small compared to the total estimated known species, if we look to these species individually, and especially if we considered near threatened species to also be of conservation concern, a little bit more than half of these species need some conservation action to reverse the situation of threat they are current in. So although we need to continue to sustain these efforts in red listing for really have a comprehensive assessment of the threat status of Brazilian fungi, we also need to urgently start to design projects and take actions to specific species that are already identified as threatened. So thinking about this, back in 2023, we had one project funded by the Mohammed bin Zayed Species Conservation Fund. This project was headed by Dr. Genivaldo Alves Silva, a postdoc at my research group. And this project was focused on Formicipora nubicula, which is a endemic species to cloud forests here in southern Brazil that is exclusively associated with Dreams angustifolia. So this fungus species parasitizes this tree species. And the main goal of this project was really to generate a more robust data set to support reassessment of Fumitiparas nubiqua that at the time was assessed as vulnerable, and we were hypothesizing that the species is actually more threatened than initially thought. So what we did was to revisit the known occurrence sites of the species and also other potential occurrence sites where its hosts were occurring, and also where the abiotic conditions known to be necessary for Fumitiparas nubiqua to occur were also present. And these basically intersected with high altitude areas in Santa Catarina state. So we actually did not find Fumitiparas nubiqua in one of its known occurrence sites because the cloud forest fragments in this region are really fragmented and degraded. But fortunately, we find a large population of the species in more conserved cloud forest fragments. So we were really able, based on these results, to classify cloud forest fragments throughout southern Brazil related to the suitability areas for Fumitiparas nubiqua. So these areas were classified as low suitability, where Dreams angustifolia occurs in a really low abundance, and these fragments are these cloud forest fragments are small, up to high suitability areas where we have high altitude cloud forest fragments, which are more contiguous and generally larger, with dominance of Dreams angustifolia. And also based on these results, we were able to reassess Fumitiparas nubiqua. This was actually the first reassessment made ever for a fungus species. So the species was initially assessed as vulnerable, and we reassessed it as critically endangered, mainly because we find out that the species was not really following its host distribution. So some environmental filters or ecological requirements were really impeding the fungus species to follow its host distribution. Because we found a much lower than expected abundance of Fumitiparas nubiqua, we adjusted its inferred total population size to be really small than initially thought, and also because of the degradation that we see on these cloud forest fragments where Fumitiparas nubiqua was occurring, a much higher population size reduction was inferred in the next 50 years or 3 generations of the species in the future. And also based on these results, really wanted to continue to update our knowledge on Fumitiparas nubiqua. So we were fortunately enough to fund another project with Mohammed bin Zayed Species Conservation Fund. And now, based on this data that we generated, we really wanted to focus on understanding better the biology of the species in these high stability areas that we classified to find sites they are essential for the species conservation to start to design in situ conservation actions, and also to establish cultures of the species to preserve its genetic diversity in a ex situ conservation approach. So this project is also headed by Genivaldo, but also Elisa is conducting this research under her master's, and sometimes we help with some field work. So what we are doing now really is to identify and follow through time, revisit every sporome of Fumitiparas nubiqua that we are able to find, and take measurements of its size, its position in relation to the ground and where in the tree the sporome is being produced, the direction of the sporome, and also some abiotic data on these cloud forest fragments where Fumitiparas nubiqua is occurring. And we are also taking notes of every biotic interactions that the species have with other fungi or some insects to really understand how the species interacts with its surrounding communities. And we are also trying to understand more of its generation length to design a method to bring more accuracy to these estimates by really trying to correlate the production of a new fertile layer in Fumitiparas nubiqua sporomes with its generation length, and with simple methods that enable us to quantify this growth. Finally, we are also trying to collect in-field spore prints to really try to isolate the species in non-invasive ways to really reduce the number of sporums that we take out of the species habitat to cultivate it. So far we are monitoring 50 individuals of these species to really collect data on all of these aspects that I have said, and also on how the species is interacting with its host. When the individuals of Dreams angustifolia die, what happened with Mitparanobicola, if it is replaced by other fungi or it continues on the substrate, and these kind of questions. And we have also 3 DNA confirmed isolates being preserved. So we are actually taking actions to preserve the species and genetic diversity in this ex situ conservation approach. We actually submitted a species pledge to the Red Accelerator Award, and we were among the finalists. So it was really nice to see that Formicola nubicola is really serving as a flagship species for fungal conservation in Brazil. And this may be indicative that we are on the right track to really try to conserve these species. In addition, or in synergy with these projects funded by the Mohammed bin Zayed Species Conservation Fund, we are also establishing the first ex situ collection of threatened fungal species in Brazil, which is funded by the Indianapolis Zoo. The project has other aspects of interactions with animals, but one of the main aspects of the project as well is to really establish these isolates of threatened Brazilian fungal species to preserve their genetic diversity. In the first culture collection dedicated to it, the Brazilian Threatened Fungal Species collection, and we are being successful and already have some isolates of some threatened Brazilian fungal species such as Bonazevia logesi and Anthrogyna tropica. We are taking similar approaches to other species as well, also in projects funded by the MBZ Species Conservation Fund, such as Rectopora araucari, where we are making targeted sampling efforts throughout its distribution range, which in this case is the Araucaria forest region in this gray shade here, but we can see that what's left from this formation is this polygon, so it's a much more fragmented and threatened biome than the cloud forest. So this species might actually be even more endangered or threatened than Formicola nubicola, because we only find it really on its previously known occurrence sites. In this project, we have also revisited some fungarium collections to really gather all data possible regarding Rectopora araucari, and based on this data, we build distribution models for the species on current climate and 2 different future climate change scenarios, a more optimistic and a more pessimistic one, to really see if this potential distribution of the species is being protected or will be protected in the future, and also to understand its intersection with the remaining Araucaria forest fragments that we still have. And this project has really concerning results, in my opinion, because most of the potential distribution of this species is really not protected in any scenario here, and also just a tiny percentage of this potential distribution is intersecting with Araucaria forest fragments. So really, if any conservation action in the next decades are not really thought and applied, this may be the first documented extinction for a fungal species in Brazil. So yeah, we should be concerned with Rectopora araucari as well. There are several other similar projects that are funded by the MBZ. Dr. Nelson Menal is working with Pleurotus magnificus, which is an edible species. Dr. Larissa Triveler is also working with Hikele dualis. Actually, Nelson is part of this project as well, and they are doing a really cool outreach in social media to really try to include citizen science in the project. And this really helped to understand the species distribution made by amateur mycologists. So it's a really interesting project as well, because she's collaborating with Argentinian and Paraguayan mycologists. So Vicalidulis occurs in other countries, and it's really interesting to see that fungal conservation in South America is being made by the collaboration between mycologists from several countries. And we have also a project focused on Bonazeve Logesi, which is managed by me and Ricardo, my supervisor, which is related with uh, constructing distribution models for several species, including Bonazeve Logesi, to really understand the distribution in the future when facing climate change. So now to finish my talk, I would like to show to you a little bit about how we are pushing changes in Brazilian environmental law based on the advancements that we have made to include fungal species in the global red list. So we currently does not have a Brazilian red list for fungal species, but this is about to change because some efforts made by the steering committee of the Brazil Fungal Specialist Group and also other members that started to join the group really pushed a decree that was recently enacted by the Ministry of Environmental and Climate Change, which was enacted in 2024, which includes fungi or the fungi in national environmental legislation. So we are really going to use the current environmental law framework from Brazil to start to conserve fungal species. And Brazil now legally has this obligation and responsibility to preserve its fungus. So this really opened the doors to Brazil start to take fungal conservation more seriously, or at least consider fungal in its conservation actions. So we are currently meeting with environmental analysts from the Rio de Janeiro Botanical Garden, which is the institution that will be responsible to publish this national red list for fungal species, and to discuss and create a partnership agreement for including fungi in this first national red list. At a more regional level, we are also being able to collaborate with the Santa Catarina State Environmental Institute to include fungal species in action plans that are designed for threatened species. So now fungal species in the Pátio Planalto Sul, which is a region that encompasses a strategic sites where threatened species occur, is now considering the threatened fungal species that occur in this area. So even without a national red list, we are being able to include fungi already in these action plans that are really the main way in which Brazil preserves and conserves its threatened species. For anyone that is more interested in reading in more detail all of these advancements, we have this recent published article, so you can read and understand more in depth all of these advancements. But I really want to thank again all of the people involved in fungal conservation in a country and are really advancing this area in a country. And yeah, thank you for your attention. I'm now open to discussions as well.
Thank you so much, Kelmer. That was amazing, the amount of work that's being done at the national, the regional, and the species specific level. And the non-invasive spore print is amazing as well. I think we should continue with Greg, but I will note there are two questions in the chat. We can address them at the end, or someone who's not speaking can respond via text as well in the chat if you'd like to do that. But I would like to next introduce Greg Mueller. If you want to get your slides ready, Greg, just a moment here. So Dr. Greg Mueller is the Emeritus Chief Scientist at the Chicago Botanic Garden and founder and co-chair of both the Fungal Conservation Committee and the Mushroom Bracket and Puffball Specialist Group within the IUCN-SSC. And ready when you are, Greg.
Great. Thank you. Thank you all for being here today. I want to kind of expand on these amazing two talks that we just had with Josefa and Kelmer, and talking about an initiative that we're doing to develop a global strategy for fungal conservation. And so I'm part of that, and then being co-led by Katia Cantiero and Rachel Hoffman. So, Kelmer kind of ruins my punchline here when I said that fungi are rarely included in national, regional, international conservation policy and action. It's true, it's rarely. You've just seen an example of where it can be included. in the work of Brazil. But that's really an exception rather than the rule globally. And an example of that is this was the logo for the United Nations Decade of Biodiversity, which went from 2011 to 2020. And on here you can see fish and birds and trees and people and butterflies and whatever else. But there are no fungi there because fungi were not explicitly, not even mentioned in documents for the Decade of Biodiversity. And this, of course, is a problem. For one thing, fungi are super abundant, and most of the people on the call know this, but just in case, just to reinforce that, this is a set of cartoons where each of the critters are drawn into scale of the number of estimated species. So for vertebrate animals, we know about 74,000 of them. We figure there's another 5,000 species of fish someplace to bring it up to 80. Plants, we know most of those up to about 450,000. But then you get to fungi and to invertebrates, where we really know only a fraction of the diversity of those species. So for fungi, we're now at about 160,000 described species, with an estimated 2.5 or more species of fungi. This is interesting, but if we do just the corollary of that, this is the same kind of cartoon, but this is looking at how many species in each of those groups have had a published red list. So an assessment of the extinction risk of their species. And you can see that, not surprisingly, most vertebrates have been assessed. A lot of plants, it's actually a much larger number now because the tree initiative is done. Invertebrates are still pretty low, but still at least 2% now. And fungi are still much less than 1% of fungi have been assessed. And this is 1% of the described species. So the 160,000 species, not of the 2.5 million species. You might have heard about this. One of the big conservation initiatives now is the 30 by 30, which specifically calls for the effective protection and management of 30% of the world's terrestrial, inland water, coastal, and marine areas by the year 2030. So it's a very inspirational goal. But Nature Conservancy, basically says it's the biggest conservation commitment the world has ever seen. Okay, great. What are we for fungi? So one of the main types of areas that are being set aside are these key biodiversity areas. They've been determined to be international important in terms of biodiversity conservation. But out of the 16,589 total KBAs, fungi are only mentioned in 4 of them, and it's not the trigger for any of them. So fungi, again, in the largest conservation commitment, aren't being incorporated. Now, there's some movement on this. There's a couple of discussions going on now to include fungi in more KBAs, but this is where we are at the moment. Fungi are not included in one of the largest conservation initiatives. CITES is the Convention on International Trade in the Endangered Species of Wild Fauna and Flora. Seems to be missing something there. So any fungi that are happen to be threatened by international trade are not protected, are not incorporated in this multilateral agreement. So why? Well, I don't have to really convince many of you here, but fungi are important, if nothing else, because a lot of people like to eat them, and nobody likes to die from them, as this Amanita bisporidea. But really, mycorrhizae play so many critical roles as symbionts, pathogens, saprotrophs, climate mitigation. Josefa talked about its role in development of medicine and industrial products. So it's just so much that fungi play. And in fact, life as we know it would not exist without fungi. And just one of the key things, because climate is such a big factor and carbon sequestration is such a factor right now, it's been estimated that fungi store about 1/3 of the carbon from fossil fuel emissions. So if we're really going to be serious about using nature-based solutions for for climate mitigation, fungi have to be part of that work. And so people are starting to get it. There's a number of new papers that are coming out how fungi will save the world. Saving the world's fungi might save us too. Great. But we um, we need to be doing something about that. We need to take that recognition and actually put it to work. So because we know that fungi are under threat, this paper came out in Science in 1991, talking about disappearing mushrooms, another mass extinction. Kilmer brought up a couple of this, that fungi face the same threats as animals and plants, habitat loss and degradation, loss of their symbiotic hosts, pollution, unsustainable use, climate change, etc. Now we're making some progress. Kilmer talked about the great progress in Brazil. Globally, there's now a little over 1,300 fungi on the red list. But if we look at that, 411 of the 1,300 are threatened. And then another 110 species are near threatened. So, over 50% or almost 50% of the fungi that have been assessed so far are in need of conservation action. So with that context, efforts are underway to have fungi explicited in the Convention on Biological Diversity and other international initiatives, including the Global Biodiversity Framework, the International Initiative for the Conservation and Sustainable Use of Soil Biodiversity, CITES, etc. And we're making some progress. Um, we've been talking about fungi. Remember, fungi is the equivalent of fauna and flora for fungi. And a number of organizations are starting to use the three Fs. Rewild and IUCN, SSC were among the first, but many others do, including Reverse the Red. There was an article in National Geographic last year talking about why fungi is just as important as flora and fauna. So that's great. Institutions, individuals are starting to use the fungi when they talk about biodiversity. But yet, as I said, most international policy and national policy doesn't include fungi in there. So one of the steps that's moving forward is Chile and the UK government, being facilitated by the Fungi Foundation, have put together this pledge asking for countries to sign on and agree that they recognize fungi as an independent kingdom of life in national, international legislations, policies, and agreements. And this pledge was introduced last year in Colombia at COP16. It was agreed that it was going to be brought up again this fall, fall at COP17 in Armenia. And at the moment, there's about 13 countries have signed on, at least last time I checked, and others are talking about signing on. But as we'll see, one of the issues is that countries say we want to do this, but we don't know how to do it. How do we incorporate fungi into our conservation work, into the commitments we've made for conserving and using sustainably and equitably biodiversity? And so one of the main legal frameworks is the Convention on Biological Diversity, the CBD. And so this agreement was ratified by 196 countries. Those of you from the US recognize that the US did not ratify it, but at least until recently, we actively promoted and followed along with most of the recommendations of the CBD. And so the most recent iteration of the biodiversity framework is the Kunming Montreal, where it was agreed to global biodiversity framework. And it calls for countries to take urgent action to halt and reverse biodiversity loss, to conserve and sustainably use biodiversity, and do that in a fair and equitable way to share those benefits. And the framework has four major goals: to protect and restore biodiversity, to prosper from the use of that, to share those benefits fairly and equitably, and then to be able to do that, to invest and collaborate so that the first three goals can be met. Now, to meet this, to coordinate action and to engage the broader plant and conservation community, the plant community developed a global strategy for plant conservation. And this was first discussed in 1999. It was actually adopted by the CBD in 2020. It's now in its third iteration. But basically, what it says is it is calling and providing recommendations on how to understand and document plant diversity, conserve plant diversity, use plant diversity sustainably, promote education and awareness about plant diversity, and capacity building for plant diversity. So some of us had this idea, what happens if we just did a search and replace and put in fungi instead of plant? And then all of a sudden we'd have a strategy that's saying we need to understand and document fungal diversity, conserve fungal diversity, use fungal diversity sustainably, et cetera, et cetera. So we thought, okay, let's see if that's a good idea. So does the community feel there is a need for a global fungal strategy to advance fungal conservation? So we had conversations with leaders from the plant conservation community, leaders in the mycological community, representing major research centers and mycological associations, and they said, yeah, that probably makes sense. So then we took it to the International Mycological Congress in 2024 in the Netherlands and held a workshop where we could really have a larger conversation with the broader mycological community to say, do we need this? And if we do, what does it look like? And at the end of the Congress, this is the closing ceremony, it was agreed on the need to develop a global strategy for fungal conservation. So the next step was we developed a pamphlet, a set of data points, talking points on the contribution of fungi to the global biodiversity framework. And what we did was we took each of the 4 goals of the biodiversity framework, and then each of the targets, because there's 23 targets that were identified to help implement and reach the goals, how fungi are needed to be incorporated in work to meet those targets, to bring up to meet the goals of the CBD. And then we took that to Colombia last year and talked to more people. And everybody's been very positive about it. So we moved forward to develop the strategy. And basically, we have the global strategy for fungal conservation now, the draft of that. It's linked to the CBD Global Biodiversity Framework. It's focused on those targets where including fungi, knowledge of fungi are essential for the success. As I said, there's 23 targets. Fungi should be included in all of them, but 11 of those targets, I think it's 11, 12 of those targets, we don't believe that can be met without including fungi. So for example, targets about climate change, how do you meet your targets on climate change when 30% of carbon is being sequestered by fungi without including fungi explicitly in that work? So it identifies actions needed to facilitate, needed to include fungi in the country's conservation policies and actions. And importantly, it's providing and developing fungal conservation and biodiversity research agenda, because we know there is more data, there's more resources that are needed to enable countries to effectively include fungi in their work. So here's the targets that we're focusing on, all the targets that are reducing threats to biodiversity. I think all the targets for meeting people's needs through sustainable use and benefit sharing, and some of the targets for tools and solutions for implementing and mainstreaming biodiversity. And just as an example, what you get is we go through each of the targets, provide what the target is, which is for target 1 is plan and manage all areas to reduce biodiversity loss. Here's the rationale why fungi need to be incorporated in efforts to plan and manage all areas to reduce biodiversity loss. A set of actions that are recommended. These are voluntary actions, but they're recommended. So integrate fungi into regional, national spatial planning efforts, but also you can include them, but you also have to do management of those. And then a series of research and infrastructure needs to be able to do the work of the actions. So in this case, refine protocols for incorporating fungi into red lists of ecosystems, identify hotspots, investigate specific fungal indicators, etc., etc. We do that through each of those targets. So here's target 4, halt species extinctions, protect genetic diversity. Again, we have the rationale, and then say, you got to catalog and map all scientifically described fungal species. You got to develop recovery plans. We need to prioritize biobanking efforts to focus on how best to maintain genetically diverse ex situ collections of known threatened fungi. And then to do that, we have to increase our efforts to document and describe fungal diversity, develop a comprehensive species profile platform so people can find the data at what do we know, develop and implement protocols to estimate population structure, dynamics, and biology about these, et cetera, et cetera. So how do we put the thing together? We pulled together a team of experts were brought together to draft each target. So for each target, we found people who we thought were experts and could contribute to the development of that target. The draft is now complete and it's being reviewed by the entire writing team, plus the advisory group and the IUCN Species Survival Fungal Conservation Committee. After we get those comments integrated into the next draft, that will be sent around to the broader mycological and conservation community for their input. And then with the goal that the strategy will be launched at the UN CBD Conference of the Parties being held this October in Armenia. So that's where we are. Just to summarize, the time is now to have fungi recognized on par with animals and plants in conservation legislation, policy, and action. Countries are expressing an interest to do this. They're interested in the pledge, but many are also concerned that the needed data and resources are not available. So the global strategy is set up to lay out the actions needed for countries and the mycological community to enable fungi to be included in policies and actions. So thank you for listening. I don't know if there's going to be time for questions, but that's what I got.
Great. Thank you so much, Greg. Yeah, we are getting up to the hour point here, but I imagine the speakers would be happy to answer questions if there are some. I believe that the Q&As that were in the chat have been answered, but if folks want to enter more Q&As or put them in the chat. I'm not sure if people can unmute themselves from my host view. I'm not completely sure. But while we give it a couple minutes here, I can ask a question or two I have. Josefa, I was actually curious, since you have this applied aspect to your project, is there a reason to think that these aquatic fungi might have more interesting or useful secondary metabolites, small molecules, than terrestrial fungi? Or is there a hypothesis that they have any difference, or are we still kind of figuring that out?
Yeah, no, I think we don't know. We are trying to figure that out. I'm interested in that question. One of the reasons I feel that they're understudied, untouched, nobody has looked at. So that's something that gives me a rationale to look into them and study them for the chemical compounds. And they're so rare, so unusual that you never know. They're so hidden that they might be having some interesting chemistry there. I know as far as bacterial antibiotics are considered, we have found some interesting compounds from freshwater fungi, that because they live in close proximity in the water with bacteria, and they're always interacting with bacteria. I mean, if you forget to put antibiotics in your media, all you get is bacteria. So that is a hypothesis there, because there's a lot of bacteria in the water, that it's likely you can find compounds that might be antibiotic, or quorum sensing, that because some organisms like MRSA get sort of adapted and resistance to antibiotics. So quorum sensing molecules are really cool because they don't attack the bacteria, but they attack their toxin systems.
Great, thank you so much. I did hear that people cannot unmute themselves. So if you do have a question, please enter that into the questions and answers feature. Um, I think I'll just ask one or two more unless we have any from the audience. But Greg, I bet people might want to know how they could follow up or how they can be informed of when there is a draft available of the strategy if they want to read it or perhaps leave a comment. Is there a plan for that yet?
Yeah, we're going to be sending it out broadly through every source we can think of. So through all the mycological societies, both amateur and professional, through every source we can. So I think you'll have a great opportunity to 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 be able to respond because we do want as many people as possible to read it, to make comments, and hopefully to buy into the work. So yeah, stay tuned. That'll come out. Like I said, we're in the process now of getting the first level of comments. It'll take us a little while to integrate those, but we're hoping by May or so that we'll be sending it out broadly to people.
Great, thank you. So broadly by May, we're hoping, we're excited. So there are two new questions that have been entered. So Shiny Singh has one. It looks like these are both kind of general, so I don't know who wants to take them. But considering the major mushroom classes, what's the best way to distinguish their substrate preference and trophic mode? Is there literature that identifies the particular terms to be used for this? And also, are these interchangeable when talking about mushrooms particularly? Does anyone want to address that question?
I mean, the tool that people use, it's imperfect, but it's the best one is like Funguild. There might be some other tools out there, but which goes ahead and just kind of lists species. But the trick is a lot of species have multiple options, right? So um, I think some of the rusts are pathogens, but they're also been shown to be orchid mycorrhizal fungi. So it's really hard to make a simple classification. Um, but yeah, I think Fungild. I don't know. Is there other ones that you guys think of, Kelmer or Josefa, that you use, or Jeff?
I think
Well,
fungal would
there are
be one.
similar, yeah, there is the fun traits as well. It's a similar
Oh,
database,
fun
but yeah.
traits, yeah. Fungild, fun traits, yeah.
Great. All right. And the next question here, I think also anyone could probably take this, but they'd like to ask, Sarah asks, apart from ITS-based identification, what other methods are available for identifying mushrooms when samples are already dried and you can't obtain a voucher number? I'm not sure about that, but for identifying dried fungi, I guess.
Well, ITS has been the barcode, but as people are using nanopore and other newer technology, they're using logger reads. So it's been extended to use ITS and I think into the large subunit. Um, and eventually we'll be looking at, whole genomes. But right now ITS is the minimum. I mean, that's kind of the barcode of choice. We also know that ITS doesn't work for every group of fungi. It's not the greatest thing, but that's kind of the default.
Yeah, and I would just say, if you go to the literature, you can still use old-fashioned morphology and see how that works out for you. There are some
Yeah.
books focusing on microscopic features, even some smaller ones. It certainly can be a challenge and a learning curve if you're starting from near zero or the beginning, but it's a possibility to try out as well. Um, I don't see any more questions. I just had one more for Kelmer, and then we can wrap up unless any of the panelists have questions for each other. We get any in the chat, but you did have some examples of a lot of threatened species from that region of Brazil, or maybe just like broadly Atlantic forests. I was curious, in that region where there are just fragmented forests, are there still species we think are native to there or endemic to there that are doing okay? Or a lot of the native or endemic fungi to that area just not abundant due to the current habitat situation?
Okay, that's an interesting question. Well, I think that some native species, I would say to the Brazilian Atlantic forest, which is arguably the most degraded habitat, ecosystem, the biome that we have here. Some of them can actually benefit from like artificial plantations and other kinds of anthropogenic pressures. Are definitely doing well. And even for those cases where the native species are not benefiting from these pressures, some of them are actually occurring in Amazonia as well, or are broadly distributed. So even though there may be some kind of negative impact of how degraded and how bad things are in the Atlantic forest, these species still do not characterize it as in any threatened category due to its spreadness and how broad it can occur in different substrates, so on and so forth. But yeah, I think that every case is different for these species.
Interesting. Thank you. And I see we have a question from Isabel. I think we should probably make this the final question.
Well, I have a
Um,
question before that,
oh, okay.
and that is
Yes.
that I know I saw that Irmgard and probably some other people have the question, where are we going to post this recording so that people can have it? I That's something we hadn't talked about, Jeff. So if you've got an idea of where, I I think we'll try to post it on the the fungal conservation committee website. Might make sense.
Yeah, I think we
I
can post it there or at least post a link there. So yes, if you search for the IUCN SSC Fungal Conservation Committee, you should be able to find uh, this recording there after a few days. I'll get that link set up. Thanks, Greg. All right, and a final question from Isabel here. So we know that many species are in need of conservation action, and yet we see increasing examples of culture collections being at risk of closing. So how can we move faster to increase awareness and save those fungal culture collections? Very good question, and maybe tough question for whoever wants to take that.
I would put it in, that's one of the reasons we highlight that in the global strategy, to actually, call on countries to recognize that, because right now, many of the culture collections are individual based. There's a researcher that has this, and as long as they maintain that collection, it's there, but it moves around. So, one of the big examples right now is INVAM, the arbuscular mycorrhizal collection was at West Virginia. While there was a arbuscular mycorrhizal fungal person at West Virginia, when he retired, it moved to Kansas. Now, when those scientists are thinking about retiring, we don't know where that collection is going. And so we need to have, I think, country-based recognition and support for these, so it's not so that there's long-term support for these collections. How to get that? I don't know.
I have a suggestion, Isabel. Every mycologist should isolate these fungi, work with chemists, find new pharma drugs, make a lot of money, and then fund our own culture collections.
That was great. I haven't heard that take before. Thank you, Josefa. Um, all right. I I do see there's another question in the chat. I don't know if anyone wants to address that in the chat. We can stay, but I think in general, I think we should end for today. And thank you all for attending and your questions. I think we can call this the end, but if anyone does want to type an answer to Shiny Sing in the chat, we can do that.
Thank you
Appreciate
for
everyone's
having us.
attendance.
Thank you so much.
Thanks, everybody. Thanks, Jeff, for
Thank you,
organizing.
everyone.
Thanks, Jeff.
Okay.
Yeah,
Thanks,
thank you,
Elmer. Thanks,
Jeff.
Greg. Thank you.
Thank you.